Hello students, today we shall discuss the next point, that is reproduction. Okay, reproduction. So let me take you on the screen so you can able to see what I am writing here. What is the name of the point? That is reproduction. Okay, what is reproduction? Reproduction is nothing but perpetuation of the living things. The new organisms are formed by following this property. Every organism have this property, that is reproduction. So as we know that this process is present in the every organism. In unicellular organisms or in smaller animals, and they also have this ability, but there is a difference between higher organism and lower organism for their type, that is reproduction type. Lower organisms normally possess asexual mode of reproduction. Okay, asexual mode of reproduction, whereas higher organism they have showing sexual mode of reproduction. Now what happens in the asexual reproduction? The organisms are formed from the parent and they are genetically identical. Genetically identical. Are you understanding? So whatever parent have the genes, the same genes is possessed by individuals also. So in asexual reproduction, genetically identical organisms are formed, whereas in sexual reproduction different genetical map on genetical forms are present from the parents पेक्षा वेगळे हे organisms sexual reproduction मध्ये तयार होतात. Okay, let us go on the next page. Now in case of asexual reproduction, come back to asexual reproduction again, because we will discuss about asexual reproduction first. Now it observed in the lower animals. In the lower animals, and these animals are following some types of asexual reproduction, and these are 1, 2, 3, 4. Which are those? The first one is fission. The second one is budding. Third one is fragmentation. And the last one is gemule formation. Gemule formation. So what is the first one? Fission, budding, fragmentation and gemule formation. These are the 4 different types. By following these types, different animals reproduce in a different manner, but all they are under asexual mode of reproduction. Let us talk about first fission. Now what is fission? Fission is, it involves the splitting of the parent organisms into two or more daughter cells. Splitting is the main concept behind this. So one parent is split into different daughter cells or daughter organisms. So in this case, parent is lost. Parent is lost in case of fission. Let us see with one example. And under this fission, there are some different types also. The first one is binary fission. Okay, binary fission. The name itself indicates binary fission means it is the simplest type of reproduction, and in which parent is divided into two nearly equal organisms. Two nearly equal organisms means, that is why bi word is used, binary fission. Then now in this form, the resultant daughter cells which are genetically identical, those are formed. They are following karyokinesis and followed by cytokinesis. Okay, let us see the words. Karyokinesis, you can write down in your notebook also while I am writing this. Karyokinesis is nothing but division of nucleus. Division of nucleus. And cytokinesis means division of cytoplasm. Division of cytoplasm. So whenever one cell having nucleus and cytoplasm, when divides into two, at that time nucleus is also divided and cytoplasm is also divided. Division of nucleus is called as karyokinesis. Division of cytoplasm is called as cytokinesis. And then two individuals are formed from these two. That is called as binary fission. And in which again simple one more type is there under binary fission is simple binary fission. Simple binary fission, and it is observed in amoeba. See, this is the amoeba. It is a parent cell, parent organism, parent amoeba having nucleus and cytoplasm. Once this division of simple binary fission starts, this amoeba is started to make notch here and this way, and the nucleus slowly started to bifurcate, even the cytoplasm. In the next part you can observe the structure must be like this. Nucleus is completely divided into two as well as cytoplasm, and finally in the last stage two new individuals of amoeba or the daughter cells, daughter amoeba are formed. That is called as simple binary fission. Parent is completely lost, two individuals are formed. Let us see the next type. Second type is under binary fission. Second type is transverse binary fission. Transverse binary fission is observed in planaria. Planaria, it is a platyhelminthes animal it is looking like this. Okay, this is animal, parent animal. Once again the process of transverse binary fission starts. It divide transversely. So the process when begins, it started to contract at the center like this. So here notches are started to develop. Still animal is not divided, but in the next phase animal can be divided into two. Like this. So there is a complete division now, and in the last stage the even nucleus has also developed in other, and complete development of new individuals take place like this. So two daughter planaria are formed, but as the division take place at the transverse position, it is called as transverse binary fission. Example is planaria. Next type is longitudinal binary fission. Third one. Longitudinal binary fission, and the example for this is euglena. Example is euglena. We know that euglena, let me draw the diagram. Euglena is a animal which is having ability of synthesizing the food material inside the body, and it has a nucleus and the cytoplasm and other cell organelles are also present. It almost look like a paramecium, but paramecium does not have such a long flagella. It has a flagella. The name longitudinal binary fission is taking place here. So in the next step you may observe that the notch formation begins here, and nucleus division also slowly develop. Two flagellas are also formed, and in the last step you can observe there is a big here big notch is formed to divide the animals, and two separate flagella, another separate flagella with the nucleus, with the cytoplasm and other cell organelles, and of course in the last part two daughter euglena are formed. Like this. This is called as longitudinal because the division take place longitudinally from the upper side. So it is called as longitudinal binary fission taken place in the euglena. The next type is in the fission now is after binary fission, multiple fission. Multiple fission, and it is taken place in the plasmodium. The example is plasmodium. What is plasmodium? Anybody knows? I think you must know. Plasmodium is a it is a, what is that, protozoan. It is a protozoan, and it is responsible for malaria in case of human being. So this is the plasmodium. How it is divided? It divide in a multiple manner. So see, this is one with one nucleus and cytoplasm. Normal plasmodium, whenever it starts reproducing inside, the cell remain as it is, but several cells started to multiply inside. One nucleus divided into so many cells with the nucleus present in them. These are the daughter cells. These are the daughter cells. This is called as seasoned. Actually, the complete cell is called as seasoned. This one is called as seasoned. Converted into daughter cell stage, and finally when the cell membrane is broken down, these all daughter cells are released outside. These are released outside as an individual plasmodium. As an individual plasmodium released. Daughter cells are released. This is called as multiple fission. So there are different types of fission we have seen. Binary fission, multiple fission. Under binary fission, we learn about simple binary fission, transverse binary fission, and longitudinal binary fission. And then we learn multiple fission as a separate type of fission. Okay, let us see the next type, fragmentation. That is the second one, fragmentation. Okay, fragmentation. The name itself indicates it fragment the animal into multiple pieces. It breaks up the animal into two or more pieces, and each one develop into individual. The example is again repeated, and that is planaria. Planaria. It was the example of transverse binary fission, as well as it is an example of fragmentation also. See, once again, let me draw. This is the original parent. Then it is divided into three pieces like this. So fragmentation takes place at two sites, and here we go in the last part. Three different animals are formed. Three different individuals are formed, and these are the daughter planaria. Daughter planaria are formed. So this is called as fragmentation. The next type is third one, called as budding. Budding, and the example is hydra. See, this is the one hydra with lot of tentacles. This is a mouth. This is the GVC, gastrovascular cavity. This is a parent. When this parent undergo reproduction, then a small bud is developed here. So this is a bud, development of initial bud. Then in the next, you can see the bud is developed more with small tentacles developed. The original parent tentacles are still there with the mouth and all that. So in the next diagram, you can observe this grows even more larger with mouth, and parent is still there, and finally in the last stage the new hydra is formed, with original parent. So 1 plus 1 becomes 2. This is the original parent, and now these are 2 with the parent. Individual is also formed. So this is called as development of bud, and the process is called as budding. When this bud is completely detached from the body of parent, it is converted into new individual. This is the new individual. So these are the different types we learned in this particular lecture. There is one more actually. Let us see that one also. That is called as gemule formation. That is the fourth type. Gemule formation, and it is observed in sponge. Everybody we know sponge is animal, and it belongs to phylum Porifera. It is present in the sea. It is attached to the substratum, particularly to the rocks in the sea, and it has a gemule for reproduction. Okay, it has a gemule. See, like this, there are very small cells are present inside, and such a type of spicules are present around. So this is called as one gemule, and when you go for study of vertical section of this gemule, you can see something like this. Cells are present, are arranged in this manner. There is one opening that is called as micropyle, and spicules are arranged on the outer side. Whenever this gemule is formed, the cells present inside are called archaeocytes. And whenever this micropyle is broken down, all these cells are come out, and they become separate sponges with the cells present inside. So this is called as gemule formation. It observed in the sponge. So these are the different types of asexual reproduction we learn in this lecture. In the next lecture, we shall study sexual reproduction in vertebrates. Thank you.